Hi, I'm here to talk to you about some of my research. I am Emily Messina, and I work on phages. So phages or bacteriophages, what are those and why are they important? So if bacteriophages, if you just think about the name, bacterio for bacteria, and then phage, eating. So you guys have talked about in your previous classes, I'm sure, if you've had Gen Bio or microbiology or A&P, you've read about macrophages, right? And macrophages are going to phagocytose pathogens. And so we've got that root in there, that macrophage or phagocytose. And in Gen Bio 1 lab, I know you guys look at amoebas, and you can, if you're lucky, you can see amoebas phagocytosing some prey. So the phage here in bacteriophage means to eat. And these bacteriophages are viruses that eat bacteria. And this right here is one of my most favorite pictures, this dramatization, because we often don't really think too much about bacteria because they're microscopic, they're too small to see. And these viruses are even smaller than the bacteria. Now this isn't to scale, this is again dramatized, but imagine you're a bacterial cell right here, and you look over that way and you just see one of your friends, is exploding, and you've got all of these phages that are popping out of it, and then one of these crazy phage monsters is coming for you, and it's about to land on you. And it really does have this shape. Not all bacteriophages look like this, but a lot of them do, where they've got this head, and this is where they have the viral DNA or the phage DNA, and then you've got a neck, and you've got these legs, and these legs land on the bacterial cell, and they attach, and then they inject their DNA through this neck. This is like a syringe. It swooshes, and it injects the DNA directly into the bacterial cell, and then that DNA takes over the bacterial cell, and it directs the synthesis or replication of the virus, and so you get hundreds of these viruses reproducing into the bacterial cell, and then they explode, and these viruses can then go on to infect other bacteria. Now, again, why are we interested in this besides the fact that it's really, really cool? So if you add up all the phages on the planet, and you add up all of the other organisms, from bacteria and archaea to cats and dogs and whales and beetles, and there's lots of beetles, let me tell you, and you add up all of those insects and all of those animals and all of those microbes, there are more phages on this planet than all of those organisms combined. So we've got lots of phages, and they're doing really important things. They help shape the microbial community. And where this community is varies based on what you're looking at. This could be the microbial community inside of you, or this could be the microbial community in a pond. Now, you can think about bacteriophages as predators, and you can think about the bacteria as prey. And we know that predators and prey kind of co-evolve. One kind of tries to up the other. And so you can think about how if you've got the predator, the bacteriophage, that's killing off its prey, the bacteria, then that prey might try to evolve resistances against its predator. So the phages act as drivers or selective pressure for the evolution of bacteria. Also, during replication, sometimes there can be an oops. When the new viruses are trying to package the viral genome into themselves, sometimes there's an oops moment, and they actually package bacterial DNA. And so this virus right here, maybe, if there was an oops, it's going to inject bacterial DNA instead of viral DNA. And that bacterial DNA that it injects into the cell could carry a new functional gene. And so these viruses can act as vectors, so not a vector like in math, right, where you've got, my cursor is causing me problems, but so not that kind of vector, but a vector that's taking something from one place to another. So this phage can vector move DNA from one bacterial cell to another, and that DNA might carry a new gene. It might carry a gene for maybe eating a different type of sugar that might help it in the environment, or it might carry a gene for antibiotic resistance, and so when you get sick, you take the antibiotic and it doesn't work. So these phages can be really, really important in horizontal gene transfer. So that means we're moving DNA from one bacterial cell directly to another. And sometimes this can have really important clinical consequences. So a lot of research has been done looking at phages in the marine environment, and this is because several years ago somebody was sampling seawater, looking for the things, and they're like, whoa, there's tons of bacteriophages here. And they've sort of continued in that line and looked at a lot of phages in the marine environment. And then slowly we've sort of caught up, and they're starting to look at phages in the soil and in freshwater environments. But we're still lagging behind there. And they've done some calculations, and they've determined that we've only actually characterized to date a really, really small amount of the phages out there. So we've only discovered 0.0002% of all the phages out there, which means you look anywhere in the environment and you take a sample of sediment or water or even air, and you're statistically guaranteed to find something new. And we know that there's so many phages out there in the environment. We know they're doing important things and we're regulating the microbial communities, but we don't know exactly how. From what we do know is they're important drivers of diversity and population size or mortality. So an example here, algal blooms. You guys have probably seen algal blooms where you look in the water and there's just this green muck. Now, this is important for a lot of reasons. A lot of people are concerned about these algal blooms. One, they're lasting longer than they normally do. Two, they're unattractive, and that might seem on the bottom of the list of things to worry about, but nobody's going to spend thousands and thousands of dollars to send their kid to summer camp if all of the ponds at summer camp are just full of muck. So this is an important economic factor, or people do not want to spend lots of money to go on vacation if, the lake that they're going to be vacationing at is full of muck. People don't want to travel to go fishing in places that are full of algal blooms. So this is an important economic concern. Also, sometimes these algal blooms can be toxic, and in that case they are harmful. And they can be harmful to dogs and other mammals. They can also be harmful to people. They can cause rashes, they can cause respiratory distress. And there's a really unfortunate acronym here. So these bacteria are cyanobacteria, so we got a C, and they're harmful algal blooms, so they're CHABs, which is a really unfortunate acronym. But these CHABs are very important health-wise and also economically important. And they're important to the environment because when you've got all this bacteria, this cyanobacteria, and it can screw up the levels of oxygen and nutrients in the environment. even though these bacteria can produce oxygen, that goes off into the atmosphere, these autotrophs, just like plants, they also use oxygen too. So they undergo photosynthesis and cellular respiration, and they can suck the oxygen out of the water below the algal bloom, and that means there's no oxygen for things like fish. And they can also strip nutrients out of the water, which makes it hard for other things to grow. So they can change the whole complexity and diversity of the ecosystem. And phages, theoretically, are supposed to control these algal blooms, right? We got a predator-prey relationship here, and so if you've got more rabbits, you've got more food for wolves, and theoretically the wolf population should grow because you've got more rabbits, and the wolves should start eating them. And we'll talk a little bit more about why it might be more complex than that for phages and bacteria. I mentioned respiration and photosynthesis. So cyanobacteria actually produce tons of oxygen. Phytoplankton, so phytolite plankton, so this is our microbes in the water, actually produce as much oxygen on this planet as all of the plants combined. So half of all the air that you breathe comes from plants, and the other half actually comes from microbes. And phages control the turnover of those microbes. And so globally, phages actually cycle 10 to the 11th kilograms of carbon every day, which is a huge amount of carbon that gets turned over. And we can look at a diagram of that turnover here. So you've got autotrophs, cyanobacteria, for example, that's shown right here. You've got heterotrophs, so this would be something like E. coli, bacteria that don't undergo photosynthesis, just for example. Now, you've got phages that infect both of those, and when these phages infect them, they replicate, and remember, the bacteria explode in dramatic fashion. And when bacteria explode, they release all of their innards. And their innards would be all of the lipids and carbohydrates and nucleic acids and proteins and all of that stuff, all of that carbon gets turned over and released back to the environment. And it can either get taken up again by other microbes, other organisms, and then they can get fed upon by other bacteria and then eukaryotic organisms like our amoebas, and then those get fed upon by little invertebrates, and then they get fed on by fish and bigger fish and so forth and so on. So phages actually turn over the nutrients for the bottom of the food web that all of us depend on. Now, all of these nutrients, when they get released, they either get fed up through the trophic levels, or they sediment out, and they get deposited on the bottom of the lake or river or whatever aquatic system that we're looking at. So these phages are really important for the environment. I hope I've made that case to you. And for students working with me, so I've worked with students in forensic science, in nursing, in health science, in biology, and we have isolated new bacteria and new phages. And one of the really exciting things is statistically, because we've looked at so few phages, you're guaranteed to find something new pretty much wherever you look. So that's super duper exciting. And then what we're trying to do in my lab is go beyond just looking at the genome, because what a lot of scientists have done right now is they take an environmental sample and they just look at all of the genomes in that sample. They're not actually isolating the phages, they're just getting the DNA of the phages and everything else in the sample, and then saying, oh look, we have all these new genes, oh look, we found a new virus, and that is cool and that is exciting. But it doesn't really tell you about how the phages work and their relationships in the microbial community. You can't truly explore function unless you have a model system. So a model system is where you've got one phage and one bacteria, and you can mix them together in the lab under controlled situations and see how they behave. So we've got a growing metagenomic database here, and we're finding new genes that we don't know what they do. And we can make hypotheses about what those new genes do. But again, until we have a model system, we don't really know for sure. So in this model system, you characterize what the phage and the bacteria normally do, and if you've got a new gene, well, you break it and you see what happens, because that's how like 90% of science works, is you say, oh, that's cool, let's break it and see what happens. So you could introduce a mutation or something in that novel gene and then say, oh, well, what doesn't work now? So we need model systems in order to really get a feel on exactly how these phages are working in the environment. And so that's what we're trying to do in my lab, is one, find new phages, but then characterize them actually in the lab and see what they do. So this is going to be part one, I think, of 3 segments. And so I'll pick up, we presented some posters at a conference with our data, and so I'm going to do the next segment on one poster and the third segment on the last poster to give you guys kind of a feel of the sort of work that we do in the lab. So stay tuned.